We will continue to discuss DNA replication, and we'll talk about the problem of replicating DNA at telomeres. Remember that telomeres were one of the three essential elements of a chromosome, a DNA end. There's a problem replicating telomeres, and we'll discuss that in detail. We'll also talk a little bit about organisms which don't have DNA genomes. They have RNA genomes, and what that means for our view of biology and what opportunities that gives us for biotechnology. But even though the genomes are RNA, we still have to replicate them. So specifically talking about the replication of the telomere sequences, that is the replication at the ends of chromosomes, we use here a schematic of a chromosome, a piece of double-stranded DNA with three origins of replication. And you can see three replication bubbles forming. When these bubbles collide, that's fine. That means you've completed that section. And since you have the leading strand here, there'll be no problem synthesizing this from 5' to 3' right to the very end of the double-stranded piece of DNA to produce a blunt end. But in the lagging strand right here, you have to jump ahead, synthesize back, jump ahead, synthesize back. When you get to the end, now you can't jump ahead enough to synthesize back all the way to the previous ending. So how are you going to fill in this region? Because we cannot jump ahead and synthesize back. The solution to this problem is a special enzyme made up of a protein and a piece of RNA, a protein-RNA complex, called telomerase. The telomerase enzyme is depicted here with the protein part being in green and the RNA component being depicted in blue. The problem that telomerase is tackling is filling in this overhang region where the lagging strand cannot fill in. It does this by actually making the template strand a little longer. This molecule of RNA that the telomerase has, it acts as a template for additional sequence at the telomere ends, putting down extra copies of the telomere repeat sequences so that you're extending the other strand. Now you can proceed on that strand that is having the issue and perform normal lagging strand synthesis. We can put in an RNA primer here and fill in this end with an Okazaki fragment, getting it to at least the distance required for the original chromosome end. The telomere repeat sequence, these fragments are repeated over and over and over again. Telomeres actually have thousands of copies of repeating base pairs. In most organisms, it's 6 base pairs, a hexanucleotide. The sequence is different in different organisms, but they function similarly. In humans, the telomere sequence is TTAGGG. The sequence shown here, TTGGGG, is from the organism Tetrahymena, the organism in which the enzyme was discovered. So at the ends of linear chromosomes, there are thousands of copies of the telomere repeat sequence, and these act as a molecular clock. Telomerase, the enzyme which keeps the telomeres long, is only active in certain cells at certain times. They are active in embryonic cells early in development, when a human is just beginning to form. They are also active in stem cells, which have an infinite renewal capability. And they are inappropriately activated in cancer cells. That's one of the tricks cancer cells use to achieve immortality. In the rest of our cells, meaning nearly all of our adult cells, telomerase is no longer active and our chromosomes are not getting filled in each time. Since you have the problem at the ends, each time a cell divides, the chromosome ends are getting shorter. This shortening of telomeres is what acts as a clock. Eventually, the telomeres get so short that a cell is no longer able to divide. Cells that are no longer able to divide are referred to as senescent. And they won't necessarily die, they can hang out, but they can no longer divide and make new cells. You may have heard that you do not make new neurons any longer. This is an example of cells that are senescent. Now, one thing to consider is how frequently would you expect a repeat to occur? How often would you expect a hexanucleotide to repeat by chance? Since you can have 4 bases in any position, you can have 4 bases at position 1, 4 bases at position 2, 4 bases at position 3, and so on. For a hexamer, that would be 4 raised to the 6th power. So you would expect any particular hexamer to occur only once every 4,096 bases. Obviously, here it's occurring every 6 bases, so that's statistically highly improbable if it were due to chance alone. Generally speaking, calculating the expectation frequencies for sequences is something that you need to know about, so you should know how frequently you might expect to observe any particular random sequence of 4 bases, or 6 bases, or 8 bases. And here's a link to an animation that shows the action of telomerase working. I've also included it in the edX webpage. Again, it's only about 2 minutes, and it's very helpful for seeing what happens. Now, it turns out that not all organisms have double-stranded linear DNA chromosomes like humans do. There are many other types of genomes. So while we've been talking about DNA replication, the more generic concept would be that of genome replication. And most of these other genomes are either bacterial or viral. Viruses, in particular, have just about any type of nucleic acid-based genome imaginable, including some that pose real problems for biological theory. Some genomes are made up of either single-stranded RNA or double-stranded RNA. This challenged the assumption that DNA is the ultimate repository of all genetic information. And also, there was no known method of replicating RNA in nature. So we'll explore a little bit more about viruses. Viruses are very simple organisms. Some can have as few as a handful of proteins. They don't have all the machinery needed to replicate their genome and put it into effect the processes which are needed for replication of the organism and viral life, if you want to call it life at all. The way that viruses replicate is by hijacking the machinery of normal cells. In this case, you have a virus with a double-stranded DNA genome. This virus infects a bacterial or eukaryotic cell. It introduces its DNA into the cell and tricks the host cell to use its protein machinery to make RNA and protein from the DNA, and also to copy the DNA for it to be packaged inside new copies of the virus. Eventually, the virus will cause the host cell to lyse, expelling thousands of copies of virus particles back into the environment. So that's how viruses with DNA genomes replicate, but there still is a problem for viruses that have RNA genomes. We've not said anything about replicating RNA, except making more exact copies of RNA molecules from templates. The answer to this problem comes from the discovery of a Nobel Prize-winning enzyme, reverse transcriptase. Reverse transcriptase is an enzyme capable of taking an RNA molecule and making a DNA copy from it. This enzyme was discovered independently by David Baltimore and Howard Temin in the late 60s and early 70s. Once you've made a DNA copy of an RNA genome, you can degrade the RNA, and you can create a second DNA strand, making a double-stranded DNA genome, which then can insert itself into the host DNA. This is just the portion in red. Now, it's possible to make RNA copies of this section, these being both the information to make the proteins needed for new viral particles and also packaged as the genome for more viruses. With the discovery of this enzyme, reverse transcriptase, it also came a conceptual revolution. The realization that reverse transcriptase violated the central dogma of biology, which was that DNA makes RNA makes protein. Now you had an enzyme which could take RNA and convert it into DNA. The ability to take RNA and convert it into DNA is also incredibly useful for biotechnology applications. We'll encounter this several times in this course, particularly in the third part of the class. Specifically, if you wanted to make copies of all the RNAs that a cell produces and convert them into DNA in order to have a library of all the expressed genes, you're going to need to use the enzyme reverse transcriptase. So I'll leave you, when talking about DNA replication, about a conundrum that we'll discuss in class, and maybe you can think about how this might occur and how you might solve it. Let's talk about the common laboratory bacteria, E. coli, and the replication of its genome. E. coli, I can tell you, has a single replication of origin. It has a circular double-stranded DNA. When it's time to replicate, it forms a replication bubble with 2 replication forts, and it starts replicating. And we know very, very accurately the speed at which it can replicate. And we can tell you that it takes 90 minutes to complete the synthesis of 2 strands of DNA to create 2 new double-stranded copies of the genome for 2 daughter cells. However, and here's the problem, E. coli replicates every 40 minutes. How can this be? How can cells divide every 40 minutes when it takes 90 minutes to copy the genome? Think about that for in-class discussion. And finally, here's the list of important concepts for the 2 videos. All of these will be tested extensively on the midterm. Thanks for listening.